Hello everyone, and welcome back to the third and actually the last day of our training program. Basically, today we will talk about splitting human embryonic stem cells plated on mouse embryonic fibroblast. And also we are going to give a summary about our training program. To begin the culture procedure, we start with confluent human embryonic stem cells, and in this slide we are showing a picture for a plate. Has two types of cells: the human embryonic stem cells, which is on the top, and the MEF cells, which is, as we can see, the spindly cells. After that, we plate the confluent human embryonic stem cells in dilution 1:6 to 1:10. The cells will take about 5 to 7 days after splitting to become confluent again. Then we prepare gelatinized plates before actually splitting the cells 2 days in advance. In order to do that, we have to use 0.1% gelatin and add it to each well of the 6 well plates. Then we add 2 ml to each well of 6 well plates, and after that we let it sit in the incubator for 24 hours at 37 Celsius. On the next day, when our gelatinized plates are ready, we can place the MEF cells, and we can do this seeding process 2 to 3 days earlier, but no more later, as recommended. Then we take 1 vial of CF1 MEFs from the liquid nitrogen. Then we let it thaw at 37 Celsius in the water bath for 30 minutes. After cells thawed, wash the cells in warm MEFs medium. After that, we seed the cells into the 6 well plate and make sure we distribute it evenly by shaking the plate gently. Then we incubate the cells at 37 Celsius overnight. On the day we are going to split the human embryonic stem cells, we prepare fresh steroid collagenase 1. And we have to note that collagenase 1 can be stored at 4 Celsius, and we can use it for only 2 weeks, no more than or longer than 2 weeks. After that, we remove all the media from the human embryonic stem cells and we wash it with 1x PBS. Next, we add collagenase 4 solution and incubate it for 5 to 10 minutes. So after the incubation time is finished, we add 1 ml of stem medium without basic fibroblast growth factor, so the stem cells can be lifted by simply pipetting the medium up and down. So in here, in this protocol, we are not using the trypsin to detach the cells from the plate. We are only using the gelatinized plate and the basic stem medium. This is the different from the other protocol for detaching. After that, we transfer the human embryonic stem cells into falcon tube, the 15 ml falcon tube, and then we centrifuge it. And this picture or this slide showing how we after we collecting the human embryonic stem cells, we centrifuge the cells and at room temperature at speed 200 G for 5 minutes. After the centrifugation step is done, we wash the cells, the human embryonic stem cells, with basic stem medium. And while the human embryonic stem cell being washed, we take the MEFs plates out of the incubator. After that, we remove the medium and wash with sterile PBS. And then after that, we add the stem cell medium. The next step, we resuspend the human embryonic stem cells with a medium, and then we add the human embryonic stem cells to the MEFs plated plate. And then we have to make sure that the cells are distributed evenly before placing the plate back to settle in an incubator. And we can do that simply by shaking the plate gently. Finally, it takes 2 days until the cells take the characteristic shape and the border appearance. In summary, mouse embryonic fibroblast MEFs are a critical biological reagent in stem cell research. It can be used as a feeder cells to maintain embryonic stem cells and induce pluripotent stems, and that's what we explained in the previous lecture. Third, MEFs can be also reprogrammed into a variety of more mature differentiated functional cell type like the cardiomyocyte and neurons. Last, the MEFs are important in regenerative medicine, focusing on biology and manipulation of human embryonic stem cells. I hope the training program was beneficial for you, and thank you.